So thanks very much. It was really great to be invited here. It's marvelous to speak to a group of people that are all so focused on consolidation. I've seen multiple different versions of the consolidation slide that I'll have in my own talk, and it's great to see that because I've never been in a conference before where anybody showed a talk like that. So it's a nice change of pace. I did want to mention that we just started this Center for Systems Neuroscience, so you may be hearing about that. That's bringing together systems neuroscientists throughout Boston University. So some of the data that I'll talk about was data gathered with in vitro recording and slice preparations of cortical structures, including olfactory cortex, entorhinal cortex, and hippocampus, and that's really what started me in this field, was looking at effects of acetylcholine in slice preparations. We also do unit recording in awake behaving rats, and we also do computational modeling of memory-guided behavior and consolidation effects. So here's an overview of what I'll talk about today. I'll start out by giving you an overview of the effects of acetylcholine, some of these physiological effects on cortical circuits during waking when acetylcholine levels are high. Then I'll talk about how low levels of acetylcholine would be appropriate to set dynamics for consolidation within cortical structures. Then I'll talk about why higher levels of acetylcholine level are necessary for encoding of new information. And then finally, I'll talk about the function of theta rhythm, possibly modulating encoding and retrieval on a faster time course. So I'll start out talking about the physiological effects of acetylcholine, since that's really what brought me into the field of working on consolidation, was starting with these physiological effects. So acetylcholine is very well known in neuroscience. A lot of the earliest, it was actually the first neurotransmitter that was described by Otto Lowy. And it's been extensively studied in the autonomic nervous system, where he first was looking at effects on heart rate. It's also been extensively studied for nicotinic receptor effects at the neuromuscular junction. But I'm focusing on the effects of acetylcholine within the brain, particularly within cortical structures that receive their cholinergic innervation from basal forebrain nuclei, including the medial septum, that projects along the fornix and provides the cholinergic innervation of the hippocampus, of which we've heard a lot about at this meeting. It also includes the nucleus of the diagonal band that projects, and the nucleus basalis, that both project to a range of different neocortical regions and provide a neuromodulatory tone that, as I'll show, we're proposing is changing the functional state of the cortex to regulate between dynamics of encoding and dynamics of consolidation. So this is sort of a main message of the talk, is that the levels of acetylcholine in a number of studies have been shown to change during different stages of waking and sleep. When I teach about this in my courses at Boston University, I always tell the students that I can look around the room and I can sample the levels of acetylcholine of each person in the room, because they correlate with the behavioral state. So if you're actively attending, your eyes are wide opening, you're furiously scribbling down notes or typing them on your computer, then your levels of acetylcholine are probably relatively high. And this was a study not done in humans, this was done in cats and rats, where they did microdialysis in behaving animals and showed if they were actively running around, you see high levels of acetylcholine in their cortex, and in their neocortex, and in their hippocampus. But as we've heard about, there's also a phenomena of quiet waking, where the animal or human is still awake, but their levels of attention are decreased, and I can see that sometimes in audiences. This is actually, most of this audience is in this state. But you can see, maybe a little bit less attention, eyes are starting to unfocus a little bit. And then in the extreme case, luckily this doesn't happen that often, but sometimes people drift all the way into this state. And it's really quite striking that when animals are in slow wave sleep, which you've heard about now plenty, these delta waves observed in the neocortex, acetylcholine levels are less than 1/3 of what they are during active waking. So it's really a very dramatic change in neuromodulatory state. So if I see one of you in this state, don't worry, I won't be mad. I'll just assume that you decided to consolidate what I was telling you so far, because that's the appropriate state to be in for consolidating prior information. So this is really one of the main take-home messages. So don't fall asleep yet, but wait till after this slide. When your levels of acetylcholine are high, physiological studies within cortical structures systematically show that the effects at acetylcholine muscarinic receptors set the dynamics to enhance the response to external input to the cortical circuits. So that's what this large arrow represents. It represents an increase in thalamic input as well as an increase in the spiking response of neurons to this external input. But at the same time, acetylcholine actually suppresses the excitatory feedback connections within cortical structures. So it's really changing the bias of the cortical dynamics to respond to the external input, which is consistent with what you, if you're really attending, you're noticing everything that I say, everything on the screen, and you're better able to encode it as well. In contrast, as you drift off a little bit into quiet waking and maybe slow wave sleep, then low levels of acetylcholine will result in a much lower influence of input on the circuit, on the spiking in the circuit, but will greatly increase the excitatory feedback within the cortical circuits. And I propose this is appropriate specifically in the context of this meeting for consolidation, though it also could enhance retrieval in certain conditions. So this is really the main message, that high levels of acetylcholine are appropriate for encoding, lower levels of acetylcholine are appropriate for consolidation. And like I said, I came into this not from the behavioral perspective. My first point of contact to this field was looking at effects of acetylcholine in cortical circuits. But it fits very well with the existing theories. So I described the role of cholinergic modulation in this paper in 1999, but really it was tapping into what was already mentioned earlier in the meeting, that Yuri Buzsaki described this two-stage model of memory function. The idea that in one stage, during waking, there'd be strong influences of the neocortex on the hippocampus, and then during slow wave sleep and sharp wave ripples, you'd have hippocampal activation driving the activity in the neocortex. So this was laid out very clearly in Yuri Buzsaki's paper in 1989, and it was the basis for Matt Wilson's work with Bruce McNaughton, looking at the changes in the activity patterns associated with different stages of sleep. So there actually are a lot of physiological effects of acetylcholine. I know this is not a physiology audience, so I've tried to pare it down. If you can, it looks like a long list, but I've actually cut out a number of effects to focus on the most relevant ones. But I've talked about how acetylcholine enhances the effective input on cortical circuits. This includes depolarization of pyramidal cells and cortical structures, as well as reduction of spike frequency adaptation, so basically making them spike more to afferent input. I talked about how acetylcholine makes feedback weak. This is due to presynaptic inhibition of glutamatergic transmission, which has been described in every cortical structure that's been studied, this muscarinic presynaptic inhibition. And I'll give a more detailed description of it, but it's basically how you go from a thick arrow to a thin arrow when acetylcholine is present. And then it also enhances long-term potentiation and enhances persistent spiking, and this could contribute to its role in encoding. So here's a very brief summary of some of the physiological data. This is data that I gathered with Eddie Barkai in piriform cortex, but it's analogous to what's been shown in hippocampus, entorhinal cortex, other regions of the cortex. So in control conditions, if you record the membrane potential of a neuron and then you put on acetylcholine, maybe with a physostigmine present, or if you put on a cholinergic agonist called carbachol, you depolarize the neurons by about 5 millivolts. And sometimes it's enough actually, it can be larger in some cells, it can be enough to make the cells spike spontaneously in response to acetylcholine. Right? So it's very clearly increasing the excitability of neurons. And then in addition, it's suppressing, so this is due to blocking a potassium current, this is due to blocking calcium-dependent potassium current. So in control conditions, a neuron, when it gets an input, it only should fire for a brief period of time and then stop. In the presence of acetylcholine, it can respond to the exact same input with a much more sustained firing. And on an intuitive level, you can think, if you're really bored, your acetylcholine levels have gone down, maybe you just can't fire your neurons very strongly to the afferent input that you see. But if you drink some coffee and increase your acetylcholine levels, you actually are making your neurons better able to respond in a sustained manner to the external input. So that's one big factor for increasing the influence of input. But then how does it modulate the feedback? So this is the effect that first got me working on acetylcholine. I was looking at synaptic transmission in the piriform cortex, which is the primary olfactory cortex, and testing differences in the excitatory feedback connection, that is connections between pyramidal cells within the cortex, compared to the excitatory input, which in that structure is the input from the olfactory bulb, bringing in odors. And in one experiment, I decided to look at effects of acetylcholine receptors, and I found this very striking effect. It was really one of the strongest, most robust experimental effects I'd seen. When I put on acetylcholine, the glutamatergic potentials that I was recording at these synapses, due to stimulation at these synapses, these glutamatergic potentials were greatly decreased. Quite a striking change. Not a complete reduction, but 50% or 80%. In contrast, up in this layer, stimulating the excitatory input to the cortex, the synaptic potentials during the same perfusion show much less of a change. And in fact, in some structures, such as the somatosensory cortex, putting on acetylcholine activates nicotinic receptors that actually enhance the thalamic input at the same time as the muscarinic receptors suppress the synaptic feedback. So it's a very robust effect of acetylcholine. So this is essentially showing the things that I just showed you, summarizing what's going on. When your acetylcholine levels are high, there's acetylcholine being released from fibers coming from the basal forebrain. These fibers often don't have specific synaptic contacts, so it looks like the acetylcholine in their vesicles is just being released into the cortical tissue, and then via something referred to as volume transmission, it's spreading through the cortical tissue and activating different receptors. So here, if it activates postsynaptic receptors, it makes the neurons more likely to spike, enhancing their response to input. If it activates these presynaptic muscarinic receptors, these are receptors that will reduce the glutamate release. So this is a glutamatergic potential. That's the standard connection. Every time you see a block and arrow diagram of the cortex, almost always the arrows between the regions, the arrows with the pluses, are glutamatergic connections, right? So that's the standard connectivity between cortical structures. But when acetylcholine is present, it greatly reduces these glutamatergic connections. That's why I would represent it as a thin arrow, reducing the strength of these glutamatergic connections. But it's selective. It doesn't affect the afferent input. The afferent input is maintained or in some cases enhanced. All right, so that's sort of the overview of the physiological effects. There's other effects on interneurons, for example, that I won't go into, but that gives you the basic story. And like I said, I started with this synaptic effect. I saw the laminar selectivity and I thought, wow, what would be the functional role of that selectivity? And that's what started me modeling essentially its potential behavioral function. And then out of the models, I started to connect to the behavioral literature that then, has connected so well to a lot of the behavioral literature, for instance, that Jan Born has been doing. And to connect it to the work that Iri Bujaki and Matt Wilson have been doing. The other thing I wanted to mention is that acetylcholine also enhances long-term potentiation. So when you don't have acetylcholine present, you can get LTP at particular cortical connections, but when you activate acetylcholine receptors, you'll get more LTP. And I want to emphasize this, seems a little bit confusing because you have simultaneously, at the same synapses, you'll have weaker synaptic transmission when acetylcholine is present. But then when you take away acetylcholine and look at it later, maybe when you've done some consolidation, you'll actually see the synapses are stronger, possibly due to direct muscarinic modulation of the cellular consolidation mechanism that John Wickstead mentioned. All right, so it causes a short-term suppression of transmission, but a long-term enhancement of the strength of synapses. All right, so that's kind of an overview of the physiology. Now I'm going to go and very specifically focus on how these physiological effects of acetylcholine could relate to the behavioral phenomena that you've been so excitingly studying and showing so many interesting results. So why would we need low levels of acetylcholine? I mean, it sounds like acetylcholine is a good thing. You want to have it, you want to, be attentive and encode information. You want to, a lot of people drink coffee in order to keep their acetylcholine levels high during waking. So why is it useful to reduce it during slow wave sleep? So the basic idea is that it could set the dynamics for systems consolidation. So, as I mentioned, acetylcholine effects on transmission have been shown in a lot of different structures. So in particular, it's been shown in a lot of different pathways within the hippocampus. So when I showed this selective effect of acetylcholine, I was aware that it had been shown to cause presynaptic inhibition in other areas. It just hadn't been studied in terms of the selectivity. So Hounsgaard in 1978 had shown cholinergic suppression of the connections from region CA3 to CA1 of the hippocampus. Valentino and Dingledine had shown this. What I did was to go in and look at this connection, replicate that effect, and then show that the direct input to CA1 is not suppressed. So it was to show the selectivity of the suppression for these feedback connections, but not the input from entorhinal cortex. I also showed the effect was at recurrent connections in CA3, but not the input to CA3, and in areas in the neocortex, showing that it was suppressing the excitatory feedback, not the afferent input. And this has also been shown in other studies in visual cortex, auditory cortex, somatosensory cortex. A number of regions show this suppression of feedback and often enhancement of thalamic input. So this fits very well with the standard modeling. This is the model now. Like I said, I've never been in another meeting where I saw the model presented, and here it seems to be starting every single talk. So it's a marvelous connection. The idea that the information is initially stored in a short-term store in the hippocampus, so forming an intermediate-term episodic memory in the hippocampus. Then during slow-wave sleep or even quiet waking, you could have greater hippocampal activation driving the neocortex, consolidating semantic and long-term episodic representations. As I mentioned, this is Yuri Bujaki's proposal that's been tested in the studies by Wilson and McNaughton, but also proposed in the McClellan, McNaughton, and O'Reilly paper. So the basic idea is that two stimuli might come in from the sensory input, A and B. They activate neocortical representations. This is transmitted to the hippocampus, activating different populations that are usually forming an episodic representation. So it might be two words represented perhaps in language cortex and neocortex, but then they're going to form selective episodic representations in the hippocampus. There's no immediate feedback because that's suppressed by acetylcholine, but acetylcholine is enhancing LTP at the same time. So it's strengthening connections between neurons within the hippocampus and essentially forming an intermediate-term association between these two stimuli. Then, during slow-wave sleep, you have a release, you have a reduction in the acetylcholine levels, great reduction of acetylcholine levels in hippocampus and neocortex, and this is essentially going to release the excitatory feedback in the hippocampus. So now you have much stronger excitatory feedback in region CA3, as shown by this thick arrow representing stronger glutamatergic potentials, stronger transmission from CA3 to CA1, from CA1 back to the neocortex. If you look at old papers by Winson and Abzug from the 70s, they actually show that during slow-wave sleep, stimulating the entorhinal cortex causes a larger feedback signal coming back to entorhinal cortex or stimulating the hippocampus. So it's been shown in vivo, though they didn't show that it was cholinergic in that case. So essentially you have these strong excitatory feedback that you would imagine just intuitively, oh, this is going to make a big explosion of activity that's going to spread back through CA1 to the neocortex. And that's essentially what was described physiologically. That would be a way of generating sharp wave ripple activity. All right, the sharp wave activity actually increases when there's low levels of acetylcholine, so low levels of acetylcholine in slow wave sleep is associated with sharp wave ripples increasing, but just directly blocking muscarinic acetylcholine receptors with atropine or scopolamine also will increase sharp wave ripple events. So it's a pharmacological result that's been shown. So sharp wave ripples, I'd like to mention, were first described by John O'Keefe in his famous book, The Hippocampus as a Cognitive Map in 1978, but it's been described extensively by others such as Yuri Bujaki as well. And I'm really kind of focusing on the sharp wave component that suggests very strong excitatory feedback from CA3 into CA1. And as Matt Wilson has shown you and others have shown you now, this is what's associated with the fast replay events, the replay of sequence. So this is during waking, you have a sequence of play cells activated, and then during slow wave sleep, Albert Lee and Matt Wilson showed that you get this replay of these same sequences. And it makes sense that it would be faster because the connections are stronger during slow wave sleep because there's no cholinergic presynaptic inhibition of these connections. They'll drive the neurons more rapidly. And so during that condition, we've done models on this where we've shown that a set of neurons in a hippocampal model, if you strengthen certain connections based on associations during encoding, then those can be reactivated. So now in slow wave sleep, you have no sensory input at all. Neocortex is not getting activated, but if you give random activation to a hippocampal network in these models, what you'll get is once one neuron crosses threshold and generates a spike, then it's going to selectively cause spikes in the neurons that have been previously associated with that neuron. So you essentially get little groups of associations reactivated together, or sequentially, the way Matt Wilson has shown. This sequential or simultaneous activation now can then drive simultaneous or sequential activation in the neocortex. These A and B fire in sequence, then A and B will fire in the neocortex, and I'm showing that this could then cause a slower change in synaptic connectivity between those associations in the neocortex. So this is the essence. It's on a cellular level describing exactly what you're showing in these box diagrams of the process of consolidation. And hopefully, you can then connect to the citations for the physiological data on these phenomena, which I'm sure many of you are aware of. So this is the idea how you go from encoding in the hippocampus when acetylcholine is present to this consolidation process when it's low. So I was very excited when Jan Born came out with these papers. It was actually, I think it was one of his students that came to me at the Society for Neuroscience and said that they were studying predictions based on these ideas about the role of acetylcholine. And so he went and studied specifically one prediction of the model. If low levels of acetylcholine during slow wave sleep are important for consolidation, then increasing acetylcholine levels should impair consolidation. And that's what they did by giving physostigmine. Physostigmine is an acetylcholinesterase blocker. So it blocks the breakdown of acetylcholine. By blocking the breakdown of acetylcholine, it increases acetylcholine levels. So this is a drug that you can actually give humans. Its effect is related, though not identical, to nerve gas. Nerve gas is a substance that directly blocks acetylcholinesterase irreversibly and causes such high levels of acetylcholine that it'll kill you. But physostigmine might make you nauseous, but it won't kill you, at least luckily at the doses that they were using. So it'll just increase acetylcholine levels. They gave an injection before sleep and they showed impairment of consolidation, consistent with the idea that you're essentially reducing the ability of the hippocampus to drive the neocortical activity. Another prediction of the model would be that if acetylcholine levels are important for consolidation, if low levels of acetylcholine are important for consolidation, you should be able to enhance consolidation by blocking acetylcholine receptors. So they did that as well. They gave scopolamine to block cholinergic receptors and show that they could actually enhance the consolidation of previously encoded information. So, and it's analogous to what you do if you go and take a nap. If you take a nap, you'll go into some stage of sleep, maybe into slow wave sleep if you're going to really get good consolidation. Taking scopolamine is very similar. In fact, scopolamine at high enough doses can actually easily put you to sleep. So it has the effect of putting you into slow wave sleep. But even if it doesn't make you fall asleep, it enhances consolidation. Now, there were another prediction of the model that I'll come to later. I won't talk about that one yet. All right, so here I've given you kind of a framework for a potential functional role of the low levels of acetylcholine during slow wave sleep, and Jan Born's data supports that on a functional level in the behavioral data. So now I'm going to talk about why high levels of acetylcholine could be useful for encoding new information. So this is something that once I saw this effect of acetylcholine, I started reading the literature, and right away I found very clear behavioral data on the role of acetylcholine in encoding. And these are really nice, elegant experiments that were done way back in the 70s, and in fact they were replicating effects that anesthesiologists knew about at the turn of the century when they were using scopolamine. And then, even 2,000 years ago, Romans were aware of these effects, not specifically with the chemical scopolamine, but with plants that contain scopolamine. So what they showed was that if you train subjects on a list of words and then give them a scopolamine injection, and then you test their recall of that list of words, there's actually no effect. right? So they're not impaired in their recall of previously stored information. But if you then give them a second list of words, here, they did about 140 words, usually subjects could do maybe 70 or so. Then they gave them a second list equal length that they studied under the influence of scopolamine. They tested their recall, and subjects on average would recall 5 words. So just like the physiological effects that I studied are really strong, the behavioral effects of scopolamine are very dramatic. In fact, I'd like to emphasize that there's pharmacological studies on dopamine modulation or serotonin modulation, other drugs that you can give humans. Even at quite high doses, they usually won't make the person delirious. But scopolamine at high doses, I'll actually show you later, completely breaks down cognitive function. So it's tremendously important for normal cognition. But so here it's showing it's essential for encoding information, but not for the recall of previously stored information. So in this, you could right away say, oh, well, this is because when scopolamine is present, you're blocking the increase in LTP. So acetylcholine increases LTP, the drug scopolamine blocks, reduces acetylcholine, affects receptors, decreases LTP, that should impair encoding. So for me, the paradox, when I found this effect on the presynaptic inhibition, I was thinking, well, why would you have an increase in LTP with presynaptic inhibition at the exact same synapses? So I'm going to give you an example in a paired associate memory task that actually formed the basis for a computational model that we published and then we tested behaviorally. So this is, you guys are all very familiar with paired associate memory tasks. You know that it involves giving a word pair. In this case, I'm giving an example of related word pairs because that's what we use so that subjects could learn lots of word pairs. Word pairs such as leather, holster, if they're presented in a particular context, are modeled as being learned by Hebbian modification of excitatory connections between a population of neurons representing this word leather and this word holster, as well as associations with the context. So then at recall, you say, what word did I just associate with leather in this context, in this, on this day, in this location? That activity would then spread across these modified connections and drive this population so that you can generate the correct word holster. So that's a standard model of associative memory function in the hippocampus. So that works very well for a single paired associate or for completely non-overlapping paired associates. But what happens when you have overlapping paired associates in this model? Then you run into problems. So we'll give the word leather combined with the word boot in a different context, as is usually done in these paired associate tasks. But now you have the problem in this model that the activity can right away spread across the previously strengthened connection and activate the word holster, which could inhibit activity for the population representing the word boot, and actually strengthen connections between the second context and this first word holster, so that when you give the word leather now in that second context, it actually activates the word holster or none. So this is what we modeled in a paper in behavioral brain research. And we showed that we can get rid of this problem by having high levels of acetylcholine. If acetylcholine is present now, you can see it's causing presynaptic inhibition of glutamatergic transmission. So when you give the word leather and the word boot, the input is not suppressed. These entorhinal input connections are not suppressed by acetylcholine. So they activate the populations, but you don't have the internal connectivity spreading, causing retrieval. So you don't have retrieval of holster, and at the same time as it's suppressing transmission at these connections, acetylcholine is enhancing long-term potentiation. So as I showed you, the suppression of transmission is not complete. It can still activate postsynaptic NMDA receptors, and acetylcholine actually increases NMDA currents and increases LTP. It increases modification so that it can strengthen the synapses. Now if you give the word leather in this second context, you can correctly retrieve the word boot. So this is why acetylcholine would be important for encoding new information, both for enhancing the LTP to store the information, but then also for suppressing the retrieval that would cause proactive interference. So that's the direct prediction that we tested. The prediction was that scopolamine, by blocking acetylcholine, should enhance proactive interference. So if we tested subjects on a series of related word pairs, 16 word pairs in a list, and then we did a series of studies, tests on this, then we training trials, and we gave scopolamine We waited enough time, over half an hour, for the scopolamine to take an effect, and then we gave some overlapping word pairs and some non-overlapping word pairs. So overlapping like leather boot, non-overlapping example would be kitchen spoon. And we tested this prediction of the impairment, of greater impairment of the overlapping paired associate retrieval, AC, versus the non-overlapping retrieval, DE. And we saw this effect, so in control conditions, their performance with saline injections, their performance on the two sets of word pairs were similar, but after learning under the influence of scopolamine, they had a greater impairment of their retrieval of these AC, these overlapping word pairs, versus the DE, the non-overlapping word pairs. So this fits then with the work from Jan Born's laboratory. Stefan Geis was one of the people that had done a lot of this work. I already talked about these studies. We showed that it enhances proactive interference. There's also studies in animals from Ray Kesner's laboratory showing that receptor blockade impairs encoding in rats. There's a number of studies that have shown this behaviorally. Scopolamine impairs encoding. He also showed that acetylcholine enhancement can impair retrieval dynamics with physostigmine. So this is essentially supporting this potential role in encoding. Now, as kind of a little bit of a digression, I want to talk about this overall dynamics. What if you go to really high doses of scopolamine? Well, you're essentially reducing the modulation of acetylcholine. Acetylcholine, when it's present, if you increase acetylcholine levels, you're going to decrease excitatory feedback. This was studied actually in Mark Desposito's lab by Michael Silver. This is an fMRI study of visual cortex. They looked at the activation with a checkerboard pattern on the visual cortex, looked at the spread of activation for retinotopic input, and then they tested that same input after giving donepezil, which increases acetylcholine levels. So some of you might have heard of donepezil. How many people have you heard of donepezil? It's also known as Aricept. It's a treatment for Alzheimer's disease because it increases acetylcholine levels, and Alzheimer's is proposed to at least involve decreases in acetylcholine levels. Though unfortunately it doesn't cure Alzheimer's disease. That was disappointing, but it does alleviate the behavioral symptoms for a period of months. So high levels of acetylcholine will reduce the spread of activity, but in the opposite direction, if you block acetylcholine muscarinic receptors with scopolamine, that means even in the waking state, you can have excessive feedback from the hippocampus to the neocortex. And this is something that, like I said, has been known actually for 2,000 years. It's been known that the various plants, including Datura stramonium, which is also known as Jimson weed, it's very prevalent in the American South. This plant contains the muscarinic blockers scopolamine and atropine. And this is useful. I mean, it was used medicinally. It was a way of getting scopolamine to use in preparation for surgery, but somewhat surprisingly, it's also a drug of abuse or a plant of abuse. So in the South, it's popular for teenage kids, young adults to go out and gather Jimson weed and eat the Jimson weed to get the effects of scopolamine. And they're quite dramatic. Like I said, it can cause a total breakdown in function. So I'm going to scroll forward to where they show these kids. Unfortunately, it can be fatal because it blocks the autonomic nervous system effects. So this shows two kids that have taken a lot of Jimson weed, and they're sitting in a police station. They don't play the sound on the tape, and the announcer had a very kind of hyperbolic tone of voice, but you can see he looks like he's passing a cigarette to his friend. His friend takes the cigarette, looks at it. There's no cigarette there. This guy taps the cigarette, smokes it a little bit, taps it again. There's no cigarette there. His hand is completely empty. This guy's combing his hair, hand completely empty. This guy sees a friend of his, motions to him to come over. Yeah, come on over. Nobody's there. You can see they're basically seeing things. They're hallucinating. They're seeing these images. He's pointing out something. The other guy tries to see it. He gets up and tries to walk. The guy says, "Oh, sit down again, sit down again." So he sits in the chair, except it's not there. So this shows, I think, very striking evidence, and I hope that you kind of remember this. If you don't remember anything else, it's pretty striking. This is consolidation gone wild. All right? This is consolidation when you don't want it, when you're wide awake, when you're dreaming in the awake state. So this is the guy that was on the Jimson weed. He says, "Oh, I can't remember anything that happened." All right, so his encoding was completely blocked, but he has these hallucinations that are probably hallucinations of prior events, you know, seeing a friend of his, smoking cigarettes, combing his hair. Essentially, they're, you know, what would presumably be going on in your brain when you're having these consolidation stages, but in this case, they're awake. So I think that's very striking. I think that is supportive, and in fact, I've been trying to figure out ways to study this quantitatively, never figured out a way. I'm sure we couldn't get it past the IRB either, but I think that's actually a pretty striking effect. So it's causing this excessive feedback, probably, and it's not necessarily just hippocampus to neocortex, it's within neocortical levels as well that this excessive feedback causes these hallucinations. All right, so another effect that I want to talk about in terms of acetylcholine enhancing encoding, in addition to enhancing long-term potentiation and decreasing excitatory feedback, it'll also increase persistent spiking activity in cortical structures. And this is very appropriate for encoding because it can be seen as a mechanism of working memory for novel stimuli. So it's not only enhancing your ability to encode what's immediately there, but it's also allowing you to hold on to information. So when I say a word to you, those words are, you know, you're holding them in your mind for a few seconds, processing it, taking the beginning of the sentence, associating it with later parts of the sentence, and that's important if you're going to encode the information. So this is an effect that was initially described by a former collaborator of mine, Angel Alonso, from McGill University. I say former because unfortunately Angel died in 2005 of encephalitis. So he tragically didn't get to see a lot of the kind of the payoff of the research he'd done. But what he had described in entorhinal cortex, which as you know provides, as Insausti showed, he provides a strong input to the hippocampus. The entorhinal cortex has very robust mechanisms for persistent spiking, a mechanism to hold activity. So most of you, when you see diagrams of neurons, I don't know how often you've seen this, but you give a current injection to a neuron, the neuron will spike during the current injection. There's a lot of spikes here pushed together because it's a large time scale. Then at the end of the current injection, you get an after hyperpolarization and no more spiking. But if you activate acetylcholine receptors during the current injection, then you'll get much stronger spiking. And at the end of the current injection, it's the same current injection here, it ends, but now with acetylcholine receptors activated, you get repeated persistent spiking activity. The membrane potential depolarizes afterwards, causes another spike, this causes further spikes, and this spiking can continue actually for up to 13 minutes was described in the paper. We actually showed it even up to 30 minutes. So it's a remarkably robust mechanism for maintaining activity within neurons. Now this contrasts with the standard models of working memory. You've probably seen models of working memory in prefrontal cortex that are using attractor dynamics. These attractor dynamics are dependent upon excitatory recurrent connections. So in simple models of cortical structures, if you have one neuron connected with excitatory connections to another neuron and then that's connected back, and then you have some inhibition to maintain stability, then when you give input to these neurons, they'll actually, this one will make that one spike, that one will make that one spike, and this recurrent connectivity can maintain both of them active for an extended period of time. But this is an alternate cellular mechanism for working memory. Instead of the neurons being connected to each other, instead now there's this internal process. Angel called it a calcium-sensitive non-specific cation current, but it's essentially a current that will respond to a spike by generating another spike. Because calcium comes in during the spike, that activates this excitatory depolarizing current to cause another spike, and so it can continue. So you can have completely unconnected neurons, but if they get input with acetylcholine present, they can maintain persistent activity. Now, in this case, if you block acetylcholine, you should have an impairment of the ability to hold novel inputs. It wouldn't impair the ability to hold familiar inputs, but novel input activity would be blocked. So we tested that. Jill McGahey in my lab did selective lesions of the cholinergic innervation, the acetylcholine innervation of the entorhinal cortex, and tested animals on delayed non-matched to sample. So that's a delay given odor. Then after a delay, they're giving either the same odor or a different odor, and they're supposed to respond if it's a different odor. Well, the animals were impaired on this task by this cholinergic lesion if the odors were novel, but then as the odors became familiar, they would be less impaired, suggesting an impairment of this working memory for novel stimuli. We also tested this in an fMRI experiment. This was done in collaboration with my wife, Chantal Stern, and her postdoctoral fellow, Karen Schoen, who's now a professor in Boston University Medical School. They did a delayed match to sample test. There was a match stimulus. Sorry, it's saying what you're supposed to do. There's a sample stimulus, then there's a delay of 10 seconds. And then in this case, the same stimulus is presented as a test and they're supposed to respond that it matched. Or in other cases, there was just an indoor-outdoor task that didn't require maintaining the stimulus over the delay. And there was activity associated with this in parahippocampal regions, parahippocampal gyrus, perirhinal cortex during the delay period. So it's when the stimulus is not present, these regions still seem to be maintaining the activity, potentially due to cholinergic mechanisms of persistent spiking. We also looked at the correlation of this activity with a subsequent test of recognition for these stimuli, and found that there was actually an association between the delay period activity. So previously, subsequent memory studies had shown correlation that if there were greater hippocampal or perihippocampal activation for the stimulus when it was present, you would do better performance on recognition later. Well, here's a case where we looked at the delay period activity when the stimulus was not present. It was a previously presented stimulus, but there's no stimulus present, but the delay period activity still correlates with better subsequent memory performance. And this is activity in perirhinal and entorhinal cortex, which are structures that have shown this persistent spiking mechanism, so could be maintaining the activity during the delay period in order to encode it. So we gave scopolamine to these subjects, and we found that the subjects that show activity in the no drug condition, during scopolamine, this would reduce the activity associated with subsequent memory in areas such as perirhinal cortex and entorhinal cortex. So there seems to be an ability of scopolamine essentially to shut off this maintained activity. So it might be impairing encoding not only by suppressing LTP, but by impairing your ability to hold especially novel stimuli in working memory for a period of time. All right, so that was sort of an overview of what effects of acetylcholine could be relevant for the dynamics for encoding. So now I'm going to switch gears a little bit, and it's nice that this actually connects to some of the EEG research that was shown here. So people have shown effects where there is increases in theta rhythm in the cortex associated with words versus non-words, but also, I don't know exactly how to pronounce your name, Fuentemilla, I think. He just showed me data showing enhanced theta rhythm associated with learning of conjunctive associations. Theta rhythm is very prominent within the hippocampal formation and entorhinal cortex in association with learning in a variety of tasks. And it's been shown that acetylcholine actually enhances theta rhythm oscillations in the hippocampus and entorhinal cortex. So this is due to this input from the medial septum. I mentioned that the medial septum provides cholinergic input to the hippocampus. It also provides cholinergic input to the entorhinal cortex. But the medial septum also has GABAergic cells. These are the inhibitory cells, you know, that when you see arrows and you have a minus next to them, that's usually representing GABAergic cells connecting to each other. The GABAergic cells in the medial septum are connecting to GABAergic cells in the hippocampus and entorhinal cortex, inhibiting them. And these cells have been shown to be rhythmically active in the medial septum during theta, rhythmically inhibiting the GABAergic cells, thereby causing rhythmic disinhibition of the pyramidal cells. And so that's a predominant theory, and this is work by Yuri Bajaki, among others, that have shown this. Also, Steve Fox, Freund and Antal have shown this connectivity and how physiologically it could mediate theta rhythm. And I was very excited about theta rhythm effects because it looks like theta rhythm effects could essentially rapidly switch between encoding and retrieval. So I've talked about acetylcholine enhancing encoding, and low levels of acetylcholine enhancing consolidation. But these effects of acetylcholine, I thought initially, oh, maybe they're really rapid. Maybe acetylcholine levels can go off and on. Well, that doesn't seem consistent with the data. Measurements of acetylcholine that can be done over a period of seconds seem to show that acetylcholine, when it comes up, it stays up for a couple of seconds. And if you look at muscarinic receptor effects in the cortex, you pulse on acetylcholine and then block it right away 100 milliseconds later, the effects still come on over 2 or 3 seconds and last for 10 or 20 seconds. So it's too fast to make the sort of constant interplay of encoding and retrieval that you need for normal function. But I think theta rhythm could provide that. So theta rhythm is associated specifically with changes in current sources and sinks within, among other areas, within the hippocampus. The one phase of theta is associated with sinks in the input layer, lacunosa moleculare, where the entorhinal input arrives, and then an opposite phase of theta, 180 degrees out of phase, you get a strong synaptic current sink for the input from CA3 to CA1 and the spiking of these neurons. So I'm proposing that this would be an encoding phase and this would be the retrieval phase within the theta cycle, and it's going encoding, retrieval within each cycle of theta. So this diagram's sort of simplifying that process. At one phase of the theta, and you have to be careful where you're recording the theta in order to know how to define the different phases, but this would be the theta recorded at lacunosa moleculare when the entorhinal input is really strong. That's an excitatory input and appears in the field potential as a negative deflection. And at this point in time, in addition to this being the maximal current sink for the entorhinal input, it's also when long-term potentiation is most easily induced by stimulating on these connections. In contrast, at an opposite phase, 180 degrees out of phase, here there's less entorhinal input, so you're at the peak of the fissure EEG, but it's the time when the CA3 input to CA1 is maximal, and it's also, you don't get LTP at that time. In fact, you're more likely to get LTD or depotentiation stimulating at that time. So you essentially have a correlation between when the maximal entorhinal input is coming in, you have maximal LTP. When the maximal CA3 input is coming in, you actually have LTD, or an absence of strengthening. And so I don't have time to go through all the details, but there's a number of studies that have supported this. One study is by Rizzuto et al. From Michael Kahana's lab, where they did a Sternberg task where subjects were shown stimuli to encode, and then they're shown a test stimulus, and they say, does it match one of the encoding stimuli? So there are different functions, encoding during some stimuli, retrieval during another. And they saw reset of neocortical theta oscillations in humans, and they reset differently, about 140 degrees out of phase. So the encoding had one phase of reset, the retrieval had a different phase of reset. This paper from Laura Colgan in the Moser laboratory showed synchronization in rats of the gamma oscillations in entorhinal cortex with CA1 during one phase of theta, and the CA3 oscillations with CA1 gamma in a different phase of theta, and they connected it to my model saying, oh, this could provide this different input of entorhinal input at one phase, CA3 input at another. And Matt Wilson and his student Siegel just published a paper, it's in press, showing that if you stimulate at one phase of theta, you can enhance encoding in mice. If you stimulate at a different phase of theta, you can enhance retrieval. So all of these data, to me, support this notion of theta phases mediating a separation of encoding and retrieval. So how much time do I have left? 2 minutes? Okay. I'm going to just briefly show, you might ask, well, what have you been doing in recent years? I've got a number of publications in recent years, and they're relevant to both theta rhythm and acetylcholine. So I just wanted to show you a taste of some of this. This is a grid cell. These are cells that were recorded by the Moser laboratory. How many people here have read about grid cells? A pretty good portion of the audience. So as a rat is running around exploring an environment, so here we have a rat performing a foraging task. We're recording units that's similar to what Matt showed. Toss little bits of crunched up cereal into the box to keep them motivated to move around. And you can see as it moves around, the firing of the neurons, this is a single neuron recorded in entorhinal cortex. You can see it's firing in a regular array of locations. And as we speed up the rat movement, you'll see that it'll look like this, where we have the rat runs around, and this neuron selectively fires in a specific array of locations in the environment. So yeah, so this we've shown actually seems to depend upon the theta rhythm input to the entorhinal cortex. So I mentioned before that medial septum input to hippocampus and entorhinal cortex seems to drive theta. This means that if we shut off the medial septum with mucimol infusions, we reduce theta rhythm oscillations. Here it's showing a spectrogram just showing a strong reduction of theta, the 8 Hertz band, after medial septum inactivation compared to before. And that reduction is associated. So the lack of theta is associated with a loss of the spatial periodicity. When theta is present, the grid cell, this particular grid cell, is firing at 4 different locations in the environment. Remove theta, you lose this spatial periodicity. As theta comes back, you get the spatial periodicity back again. So that the reduction in theta rhythm is associated with the loss of the gridness of these neurons. But at the same time, these neurons are also sensitive to the head direction of the rat. So they have both spatial periodicity and some of them have head direction sensitivity too. They lose the spatial periodicity, but they don't remove the head direction selectivity. So it seems to be selective just for that spatial periodicity. And this, I think, connects well with some of the effects of removal of medial septal modulation. So if you infuse mucimol into the medial septum, it will reduce the gridness of these cells. It also impairs their behavior, the rat's behavior, in the Morris water maze. So in control conditions, a rat can learn a hidden platform location in the Morris water maze. If you inactivate the medial septum, they are impaired. If it's inactivated during encoding, they're impaired in remembering the platform location. And I believe this could be due to loss of the medial septal modulation. It could be loss of the GABAergic modulation, but it could also be loss of the cholinergic. And we've showed, now not with mucimol infusion, that's what we did before, but more recently we've done blockade of muscarinic receptors with systemic injections of scopolamine, and we also see an impairment. So here's a grid cell that fires in regular locations. During the scopolamine injection, you lose that grid cell periodicity, and then it comes back afterwards. So that's been some of the more recent work in it, and I hope to connect back to the consolidation mechanism because now we have very robust representations in entorhinal cortex that seem to be modulated by theta rhythm or by cholinergic modulation. So I'll close there just to remind you, I showed you about how as you go to sleep tonight, you go into slow wave sleep and your low levels of acetylcholine can help you consolidate my talk. And hopefully your levels of acetylcholine were high while I was speaking. So thanks very much.